Hello, everyone. I hope everyone's caffeinated by now, and thanks everyone for attending. My name is Xiaoheng, and in case you don't know how to pronounce it, rest assured, most Chinese people don't know how to pronounce it either. I'm a postdoc with Matthias Styrokin at the University of Chicago. We do method development in population genetics. Today, I'd like to tell you a bit about this toolkit we made to serve the selection analysis of temporal genetic data. We call it Diplolocus, and naming credits to Matthias. To start off with the rapid climate change and all, we are seeing increasing interest in studying adaptation in the short term. However, before this decade, most genome data sets are contemporary. That is, the samples are considered to be taken at only one time point. Needless to say, this single cross-section of genetic variation in the population can only tell us so much about the evolutionary history. With the sequencing techniques rapidly advancing, we're seeing this boom of genetic data sequenced from fossils, museum samples, monitored populations, or experimental evolution systems. All these data sets contain additional timing information for the samples, and that allows us to analyze the genetic variation as a time series. Just to make a point in how abundant they are nowadays, although I guess it's not very appropriate to use human in an evolution meeting, where human geneticists are really privileged, just saying. Last time I checked, this AADR, Allen Ancient DNA Resource Database, David Reich's lab, have over 16,000 pseudohaploid genomes of Asian human. 16,000, yeah, that's a lot. And it would be a waste not to make good use of them. With this additional time axis, we can learn a lot more about the population genetic history. To make an analogy, it's like comparing photograph to movie footages now that photographing gets easy enough to make motion pictures, so to speak. However, despite this progress in data availability, the development of analytical tools still need to catch up. It's not like we don't have framework to analyze allele frequency dynamics. We do, and there's a whole bunch of that. It's just most of these existing methods for diploid organisms only consider selection on additive alleles. That is, dominance coefficient is 1/2. Here, just for reference, for future reference, we'll be using these two fitness schemes interchangeably, where we either use subscripts to indicate number of alleles or genotype, or following what SLIM is doing, use S to denote selective advantage of a homozygote, and use H, the dominance coefficient, to indicate its behavior in a heterozygote. And in the case of additive selection, H would be 1/2. And it's not just the dominance thing. Most of the existing framework also weren't prepared for the amount of data we have nowadays. They perform analysis on a side-by-side basis, so the computational load increases almost linearly with the number of loci. And it's easy for a handful of them, but nowadays with a genome variant called dataset easily having tens of thousands of SNPs, it's just not manageable to use old frameworks to analyze new data. So, what do we want? With all this set up, we now know that in this genomic era, we want our tool to be able to incorporate genetic time series and be able to host large number of loci and all the while account for non-additive selection. Not to reinvent the wheel, let me first walk you through how existing framework incorporate temporal samples in the analysis. Suppose on a single independent locus in this population, on this locus we take samples at 3 different time points. We can assume that in each generation, the allele frequency in the population, not the sample, is only affected by that of the previous generation, aka Markovian. Then we can construct a hidden Markov model where the hidden state is the population allele frequency as a function of time. Of course, at each time point, there are definitely way more than one possible frequency value. So rather, it's a distribution, a probability distribution for the allele frequency here. As time passes, we can use transition density function to describe influences from mutation, selection, and drift under Wright-Fisher model. And effectively, when generation iterates, we are integrating over all possible trajectories of this P of t during this time period. Then when we take a sample at this given time, say T1, the emission probability can be the binomial sampling likelihood of observing such a sample given the underlying frequency distribution at this time. We do this for every additional time we sample, and in this way, after incorporating all our samples, we will have all the overall likelihood of observing all our data under the population parameters we use. Now recall how we want our tool to be. To reduce the computational load, we simplified each transition step as a Gaussian progress, basically a normal distribution with the mean and variance being derived from PopGen parameters under Wright-Fisher model. And when parameterizing this model, we allow the dominance to vary, so to consider non-additive selection. Further, we vectorize the majority of the computation in the software such that it can compute tens of thousands of loci within reasonable amount of time, given a reasonable parameter grid. We call this diplolocus, and again, naming credits to Matthias. This is a Python package that has both programmable modules that you can incorporate in your own Python code and the command line program that you can call on the terminal. For those of you who are familiar with the Unix terminal, you can just pip install it. And it has functionalities both for simulation and for likelihood for the time series genetic data. And just to show how fast it could be, because for simulation, it would be up to your screening criteria for when a loci is discarded or redone. When we compute likelihood, for example, here are over 1,000 SNPs sampled at 9 time points, each time sampling are 20 diploids. It took around 34 minutes to compute over a 51-point grid, parameter grid, and I would say that's kind of impressive. And yeah, I'll just check off the first two here. And we know in theory that the non-additive selection is considered because it's an additional variable in our additional parameter in our framework. How does it actually perform? So we simulate loci evolving under given selection coefficient and test for selection with the likelihood ratio statistic. That is, we use the maximized likelihood over the selection coefficients and the ratio of that versus the likelihood of no selection when S equals 0. Shown here are ROC curves. The closer it gets towards the top left corner, the better the test performs. So we see it performs well not only when the initial condition used in the simulation and likelihood computation are identical. When we relax the initial conditions to be more realistic, the good performance still holds. And moreover, when we check the maximum likelihood estimates of selection coefficients, they reliably center on the true values, and which is also good. I'll spare you from many other result plots and just tell you that here, diplolocus satisfy all we need this new tool to be. But that's not everything, though. We want to know when we apply it on the real data, since we are doing likelihood ratio test, can we use the p-value out of this likelihood ratio test? So to check this, we examine the likelihood ratios of neutral loci we just simulated and compare their distribution to chi-square, which is in theory what likelihood ratio should follow. So here are the resulting QQ plots. The closer these dots are to the x equals y line, the better this distribution resembles a chi-square. And then the more reliable the p-value would be when you apply it on your data. We consider different combinations of domination coefficients computed during likelihood and the initial conditions. and found that the best combination is when we compute likelihood using a uniform initial distribution, that is no assumption at all, and fix the dominance to be 1/2, just for a speedy run. With this knowledge, we move on to apply Diplodocus on human nation DNA data. We take data from this AADR dataset and only take UK samples younger than 4,500 years old. After a few layers of qualitative filters, we ended up with 520 individuals, and with a generation time of around 28 years, there are more than 90 different sampling times. This is the whole genome Manhattan plot, and in case the color is too light to see, this is our top candidate there on chromosome 2. Further zooming in on chromosome 2, this SNP is none other than RS49AA235, which sounds very random to me, but if you look into the literature, it is the SNP that's experimentally shown to be an enhancer upstream of the lactase gene that promotes the expression of this gene. And when we further zoom in, we can also color the dots by the maximum likelihood estimates of the selection coefficients. And we can see the closer we get to the target SNP, the more significant it gets, and the stronger the selection is estimated to be, just from the darker this color would get. And note that these results are all done while fixing the dominance to be 0.5. When we know for sure that this is the target SNP, we can relax the dominance and do a two-dimensional, two-parameter search and to see what the dominance is like. Here, I plot this two-dimensional parameter space where X and Y are selective coefficients for heterozygote and homozygote. I also have the population scale selection coefficient sigma on the other side, just for reference. In this space, if the local optimum ends up on this y equals 0 line, that means heterozygotes have 0 selective advantage. In other words, it's completely recessive. Likewise, any points on this x equals y line would be full dominance, and on this red line would be y equals 2x line, it would be additive selection. Then anything in this red area would be slightly recessive, and in this blue area would be overdominant. So this is what we have on the data of this lactase SNP. We obtain our local maximum at 0.05, 0.07. It's close to previous estimates, and it's also consistent with experimental evidence that show comparable enzyme efficacy as long as the beneficial allele is present. So to quickly summarize, we have gone through how this framework works and why it works well. We show in simulation that it performs well both in computational speed and statistical performance. And we apply it onto human nation DNA data, yielding results consistent with previous knowledge. For the sake of time, I'm gonna skip through the next part real quick and just tell you it can also be applied onto pulse-seq data, pulse-seq allele frequency data. With that, I'd like to thank my advisor, my colleagues, and funding sources, and happy to take questions.
Really cool stuff. I'm wondering if you could go back to the lactase locus zoom plot, because you had some blue dots there too that were next to it. I'm just wondering if there's a sign. Back one or two more.
Oh, this.
Yeah. I just noticed that you got negative values in blue.
Yeah, they're linked to the other allele.
Okay.
Yeah. They're linked to the other allele, and actually I have a plot for their allele frequency. You can see there's a bunch that go up all the way with the solid line is the target SNP. There's a bunch that go along with it, and the other half is the opposite. Yeah. Cool.
All right. Good morning, everyone. My name is Justin Conover. I'm a NSF postdoc at the University of Arizona, and today I'm going to tell you about how to improve read mapping and SNP calling through the use of a variation graph pangenome. So first I want to start with just a very basic, typical population genetics pipeline. You would start with assembling a reference genome for your species of interest, or if you're lucky, there's already a reference genome available. And then once you collect some population level data, you can map those reads onto your reference genome and call SNPs and do population genetics. So for example, if you're interested in Brassica oleracea like I am, or cauliflower, you could use a site frequency spectrum like this to infer different properties of the demographic histories, or maybe a distribution of fitness effects. Or if you were interested in its sister species, Brassica rapa, or turnips or Chinese cabbage, you would take the reference genome from Brassica rapa and map reads onto that reference genome and do SNP calling generally. And in case there aren't any people who are familiar with the messy data of plants and wondering why the site frequency spectrum for Brassica oleracea looks so comb-like and so jaggedy, it's because this species is predominantly selfing, and selfing increases homozygosity, and when taken to the extreme, you see even allele counts more often than odd allele counts, which will come up throughout this talk as well. So after you have these really high confidence SNP calls, you can then do population genetics of any flavor that you choose. But if you want to learn something about both of these species together, like for example, is there a bottleneck associated with the speciation event of these two, you have to map all of your population reads onto a common reference genome. And so this creates, and you would use a two-dimensional site frequency spectrum like I'm showing here on the right, where instead of a histogram or a line plot, this is showing a heat map of which, or of how many positions in the genome are co-segregating in both of the populations that you're interested in. And so this creates a question of which reference genome would you use? And so I was lucky in that there were reference genomes already available for both Brassica rapa and oleracea, and so I decided that I would just map these reads to both of the reference genomes and decide which reference genome would be best to use. So here I'm showing data from the Brassica oleracea population, and in green is the same data that you saw on the previous slide, and in purple is the site frequency spectra that I generated when mapping the reads to the Brassica rapa reference genome. And I use BWA for mapping and GATK for SNP calling, which are both very standard toolkits in population genetics. And what you can see is there's a big difference between these two site frequency spectra. And this isn't just a small difference. You would infer a radically different demographic history based on these two site frequency spectra, which means that you wouldn't be learning anything about biology. This would just be a statistical noise, and you couldn't infer anything about the true biology of these populations. I initially thought that maybe this was a technical artifact, so I used a different SNP caller. I used BCF tools and saw even worse performance. You'll also notice that this site frequency spectra is a little less jaggedy or less comb-like than GATK, which is due to some technical differences between the two SNP callers that I'm happy to discuss afterwards. But overall, we see generally the same patterns, where there's a lot of alleles that are shown at intermediate frequencies, and again, you're just not going to learn anything about the biology of these populations with these SNP calls. So maybe the problem isn't with the SNP caller, but maybe it's the read mapper. And so instead of using BWA, I also use a program called VG. And even though it looks like these data are better, there's still a lot of discrepancy, and they're still not that overlapping. There's still really severe differences between these two site frequency spectra. And maybe a better way to quantify this is, instead of looking at the frequency spectra, looking at nucleotide diversity estimates. So here I'm showing the nucleotide diversity when using BWA or VG, and whether we're using BCF tools or GATK for the SNP calling. And notice that the y-axis here is a log scale. So the average nucleotide frequency here for every exon is indicated by the dots. And this is a twofold difference in estimation of the nucleotide diversity, which means like you can't learn anything about these populations with this messy data. The same general principle holds true for the other population, Brassica rapa. Even though these do agree more with each other, you can see that there's still some really important differences, especially in those rare variants when using like GATK and BWA in the upper right. And if you look at the bottom left corner, you'll notice that there's only a purple distribution, and that's because there's a green distribution at 0 all the way across, because we just couldn't find any SNPs that were located in every individual. And again, we can also look at nucleotide diversity here and still see that there are big biases in the estimations that we're finding. So this means that neither one of these reference genomes can be used to learn anything about these populations. So I had to reframe the question. Instead of asking which reference genome is best, I asked, well, what if we can map these reads to both of these reference genomes simultaneously? And what this means is going into the realm of a pangenome, or specifically a variation graph pangenome. You can think of a variation graph pangenome as just a fancy way to represent a sequence alignment. So here on the right-hand side, I'm showing two sequences shown in blue and yellow. At the top is just the pairwise sequence alignment, and in the bottom is how you would represent that alignment in a graph form. So each node is consistent of nucleotides, and then you can connect nodes together to form to reconstruct each of the sequences from the graph structure itself. So say if you have an indel in your sequence alignment, you can show that by skipping a node as the second to the left node is shown, or you can show a SNP directly in the middle, or you can even show an inversion through tracing that blue haplotype to the opposite side of the node and then tracing it back around. And you can actually map reads to this reference genome using that program VG that I mentioned earlier, and then you can tell the program that you want to, what's called surjecting those reads back onto one of the two haplotypes, and then do SNP calling just as you normally would against a normal linear reference genome. So I created a whole genome alignment between the Brassica rapa and Brassica oleracea reference genomes that's shown here. And I use AnchorWave for this alignment. That's a program that's specific for plants to deal with the high TE content and high repetitive regions and polyploidy that's present in plant genomes that may not be present in mammals or other model systems. There are other tools to build pangenomes like this, like PGGB, or the Pangenome Graph Building Toolkit, which may work for other systems, but it did not work for these plant genomes at all. So in the top here, each color represents a different chromosome, where the top 10 colors represents the 10 chromosomes from one reference genome, and the bottom 9 are the 9 chromosomes from the other reference genome. And the edges at the bottom show all of the connections between the different nodes in this graph. Now I will notice that because this is so zoomed out, you're not seeing most of the variation here. There's a ton of variation that's really shallow in this edge graph, but you just can't see it because we're zoomed out so far. So whenever I map reads onto this pangenome reference and then surject them back to one of the diploid references, these are the site frequency spectra that I see. Where on the top two, these are data that you've already seen before, and on the bottom is mapping these reads to the pangenome. And what you can see is that for the most part, a lot of these differences have improved. This still isn't a perfect method, but this is at least a lot closer site frequency spectra, regardless of which reference genome you're mapping these reads to. And we also see an improvement in nucleotide diversity as well. So on the left is the really biased estimations for pi that I presented to you earlier, and on the right is the improved estimates for nucleotide diversity for this Brassica oleracea population. And again, we can see the same thing for the Brassica rapa population as well, where even for that combination of using BCF tools on your linear reference, where there is no green site frequency spectrum, you do recapitulate the correct site frequency spectra using these variation graph pangenomes. And you also see a much greater improvement in the estimations of pi as well. The interesting thing is that I thought that this would be due to an increased amount of read mapping. But when we actually look at the percentage of reads mapped, we don't see any difference at all. So on the left-hand side here is showing the remapping percentages using BWA or VG on the linear reference genomes. And this is for the Brassica oleracea population. And on the right is using the pangenome reference. And so if you just look at VG on the left and the pangenome on the right, for those reads that have a mapping quality over 20, there's not much difference between the linear reference and the pangenome reference, which was a little bit confusing to me. But I think what is happening is that the reason why read mapping is so complicated in these two diploids is because they actually share an ancient whole genome triplication event or a hexaploidy event. And so there's just a ton of repetitive regions in these genomes. And when you're trying to map reads from a distant related reference genome that has a bunch of duplicated copies, it's really hard for any read mapper to know which copy of any given gene you should map a read onto. And so not only may this method be useful for species that have an ancient polyploidy event in their past, but it can also be used for neopolyploids as well. So even though these two species diverged around 2 to 5 million years ago, around 5 to 10,000 years ago, there was a hybridization event that created an allopolyploid with twice the number of chromosomes, And there's also homologous exchanges in this polyploid, meaning that you can't even tell which chromosomes come from which diploid parent in some cases, because each chromosome is a mosaic of the two diploid progenitors, which makes these polyploids extremely hard to study. So even though these pangenome graphs can improve read mapping in these polyploids, the standard SNP calling tools don't work in allopolyploids very well. Here I'm showing GATK and another tool that's designed specifically for polyploids, but these site frequency spectra look really weird. There's a peak right in the middle that shouldn't be there. That's all due to SNP calling errors. And the peak on the right of each of these plots should be exactly at 50, because that would represent the divergence between your two diploid progenitors. But in this case, they're both shifted to 49, which means that you can't even infer an accurate demographic history or much from these data. And so to improve the SNP calling in these allopolyploids, I worked with David Gerard, who's a statistician at American University, who had previously developed this R package called UpDog, which is specifically for genotyping polyploids. And we created a new Bayesian prior that I'm happy to discuss afterwards if anybody's interested, but we can get a more biologically realistic site frequency spectra here. And you can see that there's a lot of inbreeding in this polyploid as well, which may also be contributing to why SNP calling is so difficult with these other bioinformatic tools. Finally, if you're interested in inferring demographic histories, one of the labs that I'm in is conducting a genomic histories inference strategies tournament. It started 2 weeks ago, so there's still plenty of time to enter. There's 4 different data sets that you can download and infer the demographic history any way that you choose, and just send us the results back. All of these results are due on October 18th, and there's 4 different levels of more and more complicated demographic histories. I will say, much to my disagreement, there are no polyploids in this data set. Everything's diploid. And so these are like realistic demographic histories to infer. And so you can go to the website ghi.st if you're interested in learning more about this program. And I also have some flyers up here if you're interested or want to take one to share with one of your colleagues or friends. I have a lot of people to acknowledge, especially all the people in the Barker Lab and the Gutenkunst Lab at the University of Arizona, and David Gerard at American University. And I'm happy to take any questions if you have them. Yeah.
Have you done any, or have you looked at any of the pangenome-specific callers, like with PG?
Yeah, so the question was whether I looked at specific pangenome callers. I have not yet. I think so. The callers that are designed for pangenomes mostly do structural variation, and that structural variation has to be in your pangenome graph initially. And so, like for SNP calls, you would have to have every SNP in your pangenome graph in order to genotype those. I think that's an active area of development where they're trying to do de novo SNP calling on those pangenome graph structures, but I haven't tried out those tools yet.
So my second question was, have you tried iterating, like calling SNPs and then taking that BCF and putting that into your pangenome and do like a second iteration to see if that improves?
Yeah, I haven't tried that yet. There is some discussion about ways to do that. The issue is that if your first round of SNP calling are still of poor quality, your next round is going to look really good, but it's just because you've already incorporated that bad variation into your graph. And so it's harder to know how to score the quality of those remappers as well. But that's a good question. Yeah.
What's up with CAT-K? Like, did it look like you could, like BCF tools with the pangenome resolve some of those spectrum frequency that you're looking at? But CAT-K kind of kept like the awkward distribution. So what do you think is kind of being happening there, and why the different callers are having different effects?
Yeah, so it was specifically the low frequency alleles that were the big difference between GATK. And GATK uses a lot of properties about like the read mapping quality or the base quality scores of the data, whereas BCF tools just cares about read counts for the most part. And so the input data that we had was lower quality for the Nebraska Rapid data. So I think it's a data quality issue, not necessarily a difference in the, or a fault of GATK per se. Okay, thank you.
All right, everyone. Thank you for coming to my first professional talk. I'm TQ Smith. I'm from Penn State University, and I'm a member of Zachary's Speeches Lab. And today I'm here to talk about my first project, Lodestar. Now, I like to motivate my project with Novembre's 2008 paper, Genes Mirror Geography in Europe. So what Novembre did is he took a set of European individuals and genome-wide markers, computed the PCA plot on the genotype matrix, and said if we scale out this genotype matrix, reflect it, rotate it, and shift it, we could see that the PCA plot structurally reflects the geography of Europe. Now, the key thing that I said there, though, is that he did this using genome-wide markers. And we know, though, that evolution treats different regions of the genome differently. And to illustrate what I mean by that, let's consider 99 CEU individuals from the 1,000 Genomes Project and their second chromosome, and take 2,000 non-overlapping windows. And we see that as we go along this chromosome, the relatedness of the individuals in two-dimensional space changes. So sometimes they're more grouped together, sometimes they're more spread out. Some windows look alike, some windows don't. So the really goal of Lodestar is to identify these windows that are interesting or different. But we're going to do that with respect to the genome-wide plot of relatedness. So again, I took the 99 CEU individuals and took genome-wide markers and computed their plot of relatedness. And we get something like this. And what we're going to do is we want a method to compare one coordinate system to another, but we want to make sure that we keep the structure of either coordinate systems, the structure of either plots the same. So we want to either scale, rotate, or shift one plot onto another and see how well they structurally compare. And we're going to do that using a method known as Procrustes analysis. So what Procrustes analysis does is it's going to take this genome-wide plot in red, and we're going to take a local window in green, and we're going to say, if we rotate, scale, and shift it, how well do they structurally compare? Now, what we're really doing here is we're trying to minimize the distance between each corresponding sample in the local and global window. And as we minimize this distance, we can gain a Procrustes statistic, which ranges between 0 and 1, 0 being not dissimilar and 1 being exactly dissimilar. Now, we can do this, and what Lodestar does is we do this throughout all the windows of the genome. So again, sticking with our 99 CEU individuals in the second chromosome, we have a log scale T statistic, our dissimilarity statistic on the y-axis, and the windows along the x-axis. And we have highlighted on the left the least significant windows, and highlighted on the right the most significant window. Now, the most significant window on the right, all the samples are closely related. And this window is in the LCT region. And we know that CEU individuals are European, and in European populations, the LCT gene is under heavy positive selection. They all share the same alleles, and they're all closely related. So essentially, Lodestar was able to extract the LCT gene or region. Now, you could say that either of these green points in either plot, to me, they're both very structurally dissimilar as reflected in the T statistic, but you might say, they're not really different, they're just kind of blobs. How can we really evaluate their actual dissimilarity? And we can do that using a permutation test. So what we're going to do is we're going to take our local window and our global window, and we're going to randomly permute the labels of the local samples corresponding to the global samples and recompute our statistic. So essentially what we're asking is a random configuration of points more or less dissimilar than the observed set of points. And we see that the plot on the left, almost every random configuration yields a more dissimilar plot than the one on the right. So this gives us statistical evidence that the observed T statistic in the right plot, the most significant region, is in fact significant in structural dissimilarity. Now, you may ask, why aren't we just running the permutation test on every single window? And the reason for that is twofold. The first one is permutation tests do add a little bit of computational complexity, but also I want to stress that the T statistic is actually rather informative and that the permutation test is conservative. So I use it really to evaluate the significance of the dissimilarity of individual windows. And that brings us to summarizing Lodestar's pipeline. Now, it's, I think, rather straightforward, and as you can see, I get confused between C, C++, and Python, but I hope that the pseudocode is rather straightforward. But the one thing that I do have to mention is that whereas Novembre did PCA on the genome type matrix, we're actually using a genetic distance, and here It's the mean number of differing alleles between samples. And the reason why we don't use PCA and a genetic distance and then use MDS is we could actually switch out our genetic distance function if we so desire. So we take windows, they can be overlapping, and we slide along the genome. For each window, we calculate the distances between samples, perform CMDS to get our dimension-reduced plot, and we do this for the genome-wide as we slide through the genome. And then for each of these plots, we compare, using Procruste's analysis, the local window to the genome-wide. And you can optionally do a permutation test. And with that, I would like to further enforce lodestar to extract regions under positive selection. So again, consider the 99 CEU individuals, but in addition, 107 YRI samples from the 1000 Genome Project. And we see here this region highlighted in green. And what this is with this elevated T statistics, this region contains all your HLA genes that belong to the MHC complex, which are under positive selection. Now, I do want to point out that I only use very small sample size, more for illustrated purposes. The distance calculation is quadratic in time, MDS is cubic. I wrote the program in C with a background Fortran library, and I multi-threaded it, so even with a few thousand samples, lodestar can still perform. And with that, I would like to now switch our attentions to the ability to identify inversions. Now, before we could go to inversions, we can actually hypothesize what we're about to see, right? So we have the T statistic is really a measure of relatedness or dissimilarity. Now, if we have a region that's inverted, it's going to be bounded by 2 breakpoints. We know that inversions suppress recombination. And if recombination is suppressed, the relatedness within the inversion is going to be different than outside of the inversions, and it's going to be bounded by the 2 breakpoints. So we can hypothesize that in our resulting graph, we can see that the T statistic is going to change where the inversion is. So this is a simulated 100 megabase segment with a 10 megabase inversion in the middle, and lodestar is able to essentially pull out the inversion with the breakpoints as boundaries. Now, simulations are rather idyllic, so we're going to test and see how well lodestar performs under something a little bit more stressful. And what we decided to do is use the 3rd chromosome of Drosophila pseudoobscura for all the Drosophila people here. We took we created using the left and right an balanced isochromosome, which I just would call chromosome 3, and we took, pseudo obscura from 6 different populations, where these arrows here are roughly where the inversions are. So there's 6 inversions, 12 breakpoints, and I ran it through lodestar. Now, the thing that I want to stress is that we had 6 different populations, and they weren't all equally represented in samples, and we only had 54 samples. But even with these restrictions in this graph, we can see that the green lines actually correspond to the actual breakpoints of the inversions. Even though we're not able to recall every single one of them, and they're a little off, scanning from left to right, the left side of the graph, the green lines are roughly around where the T statistic changes, and as we move along, we see that these green lines are near the shifts in the T statistic. And with that, I would like to thank my lab and our collaborator, Dr. Stephen Schaefer, for helping with the Drosophila data, Penn State for having me as a grad student, and then if you're addicted to 3 blue, 1 brown, like I am, I use this same software to do the animations. So with that, I'm a little early, but if any of you have any questions, that'd be great. Yes.
2 questions.
Yes, sir.
Where can we find the pipeline?
Oh, so I'm currently still testing it. It is currently on GitHub, but I'm hoping to have a preprint out relatively soon. And it's just a program. It's a precompiled executable, or you can make it yourself. And there's no dependencies, so the installation won't have an issue. Yeah.
And then I was wondering, could you comment on how this is a little bit different from something like PCA DAPT, where you're using Mantelodus distance to infer some of these sort of similar things, but I was just kind of curious if you have like a way of sort of explaining the difference.
Jog my memory. That's Ralph from 2017, right?
Yeah.
Yeah, okay. Yeah, so There are subtle differences in this. So what Ralph did is he, I believe, he took the distances between windows with the covariance matrices with using PCAdapt, and computationally they're a little bit different. I believe that this is a little bit more flexible, and that the pattern to specifically extract inversions, I think, is a little bit more clear with the change in the t statistic. Yeah, thank you. That's a good question. I'm currently looking into that. And it's nice to hear from you. I've read your papers. Yes. That's actually a great question. So right now, you do this visually, and I would rather do this than the alternative sometimes is people will actually just look at the alignment tracks in a genome browser and just try to visually identify this. This is a little bit clearer, but the reason why you see like just kind of that random breakpoint there is because the population representing that breakpoint in inversion is low. So I believe that it's only about 3 samples. I hope that answers your question. Thank you. Yes, sir.
Did you test that with marker samples?
I'm sorry?
Have you tested this with marker samples?
I have not. On the to-do list. Yes.
I'm just curious how long it takes to run something like this.
So that really depends actually on the number of samples. The MDS is not an issue. So if you have a couple thousand of samples on multiple threads, you can crush a chromosome in about 10 minutes. Yes. All right, anyone else? Nope. Okay. Thank you all.
All right, well, happy Monday. Thank you all for being here today. My name is Jacob Green. I'm a PhD candidate at the University of Rhode Island. And today I'm going to talk to you about our development of a tool to sequence the exome of any organism, but more specifically about how probe and gDNA library length impact exome capture sequencing within a non-model system. And so first I'm going to start off with acknowledgements. Science takes a large team, especially with our unruly oysters. And really, Amy Zick, my fellow grad student, has allowed me to piggyback on her kind of broad-scale Narragansett Bay experiment and let me tear apart the molecular methods of it. So that's been really fantastic. And also undergraduate Hector Santiago, who's been able to help me a lot with some of the DNA extractions and the library preparations, as well as the conference organizers, NSF for funding, and then also our graduate union, who has a really strong presence at the university and supports grad students and graduate assistants at URI. All right, so I'm going to start off with a controversial statement that really genomic resources and tools for model species is fairly abundant, right? I don't think we have to narrow that home, but it's changed our approaches in many ways to how we understand evolution and disease. But I really argue that it's the targeted approaches that have allowed us to really expand our analysis and really improve our results, especially in areas like human disease and cancer. And, although I'm an oyster guy, I've always enjoyed these rich genomic stories of researchers kind of reaching into the past and telling stories of our close hominid ancestors. And in 2009, researchers presented this new method of targeted ancient DNA sequencing retrieval that utilized the known Vinja Neanderthal genome and targeted specific regions within the mitochondria to better recreate that mitochondrial genome. And this allowed for greater reduced sample destruction and also lessened the sequencing demands. And so this gave us great insight into their population size relative to kind of modern humans and our great ape relatives as well. Fast forwarding to 2024, we now have organizations like Deciphering Developmental Disorders that has utilized whole exome sequencing in order to help doctors better understand the genetic cause of human developmental disorders. And so recently, DDD took 12,000 children in the UK and identified 285 genes linked to childhood ailments, including 28 new genes linked to these disorders. And still, despite this incredibly massive effort, many of these de novo mutations in protein coding genes remain unexplained, and even modeling suggests there can be over 1,000 related genes that are still yet to be identified within these disorders. And so still, targeted and whole exome sequencing have been an incredible benefit for studying human disease and evolution. But when we shift our focus away from our models, our humans, and we begin to consider species that are really the most vulnerable to climate, disease, and extinction, many of these are non-model organisms that have really few or no genomic resources available. And it is these non-model species that need targeted whole exome approaches for us to truly understand their genetic diversity, disease, susceptibility, and pressures to selection. So if your aim is to investigate these genes under selection in non-model species, I think I have a tool for you to utilize. Our lab has been developing a targeted exome capture approach as really a new way to assess these non-model exomes. And that's called expressed exome capture sequencing, or EEC-seq for short. And one of the really unique aspects of this technique is that your organism itself is creating the capture probes that are needed to target the exome. But before we dive into the tool, let's take a step back into the genomics classroom and kind of review what an exome is. So in every genome, there are genes, and these genes have introns and exons within this gene. And it is these exons that are transcribed into mRNA and eventually translated into proteins, which do work in the cell and interact with our environment. And it is these entire collection of exons within the genome that we term the exome. And by profiling the exome, we can really better understand selective pressures and mutations within these genic regions. And so how does EEC-seq actually work? Well, we take mRNA libraries, and they're synthesized into biotinylated capture probes. And so it is this way the probes actually reflect the mRNA profile of an organism. And in studying a specific system, we can create probe sets that are unique to subpopulations. These probes can be unique to tissue types, and they can also even be unique to disease exposures as well. And since we don't need a reference or genome coordinates for any of this probe creation work, we don't decide which exons are significant. Our organisms are telling us what is significant. And so we can expand this technique to understudied species such as tropical amphibians. Probes can be made not only from single individuals, but from whole communities of organisms to reflect the holoexome of a group. And we can even create probe sets from different life history stages, such as larval oysters, and use those larval probes to enrich adult exomes to explore how expression patterns early in life history translate to adult exome selection. And my fellow graduate student, Amy Zick, did a great presentation on utilizing EEC-seq and environmental variables in Narragansett Bay to detect outlier SNPs important in adult connectivity under larval selective pressures, and she's more than happy to talk to you about any of that. But some of really the benefits, just to hammer home some of this, is that it eliminates the need for reference and human bias when we're making these decisions. Next, it really reduces the cost and time. Think about how much time it takes to make a genome, extract coordinates, figure out what's significant. That's not needed within this method. It can produce thousands of exome SNPs that are situated in exons or near and within genes, so we know that they're in some way or shape significant. And it can give us some information in terms of improved understanding of gene regulation, because these probes are coming from expressed mRNA profiles. So some things will be highly expressed, we'll get probes, and if they're lowly expressed or not, we won't actually be sequencing those regions. And so my project really has been focusing on making EEC-seq more approachable and a little bit of a cheaper method. And it's in 3 phases: molecular protocol optimization, so just trying to break as much stuff I can in the lab bench. Comparing different sequencing approaches, such as EEC-seq, the whole genome sequencing, or RNA-seq, and commercial capture probe design. And then lastly, kind of a full bioinformatic development to de novo assembly exome reads, because typically you just assemble them to a genome, because that's where you're getting them from. So we need a way to actually assemble this stuff. But today, I'm really going to be focusing just on molecular protocol optimization. Just real riveting, fun stuff, right? So in order to do this, we're going to be taking a look at probe and gDNA library size. And some of our thoughts behind this is that our longer probes may capture more specific gene regions around there by increasing the length of the region that's being captured. And that longer gDNA will increase coverage across these exons as a whole. So to give you some insight into our design, we have probe libraries and gDNA libraries, each with 10 individuals within 5 subpopulations and 2 individuals within each of those populations. And our first design is lovingly called Tiny, and that is going to be our 150 base pair gDNA size and 150 base pair probe size. Our next combination is where our gDNA is larger and our probes are a little bit smaller, and that's going to be called Goldilocks, a little bit of foreshadowing throughout the presentation. Next, we're going to have our probes being really large at 300 base pairs and our gDNA being smaller. That's going to be called Biggie Smalls in honor of late great rapper. And lastly, it's going to be a 300 base pair gDNA size and 300 base pair probe called Andre 300. All right, so I'm going to be using these terms instead of confusing you with a bunch of numbers and whatnot. So once we get these reads, we go through a pretty typical read quality check, processing the reads in terms of normalizing the reads between captures, and then into a pipeline called Docent, which is largely just mapping our reads. Because we have a reference oyster genome, we can naively create these probes and then check how we're actually capturing these exons. And for mapped reads, we're really focusing on 2 areas: capture specificity, whether we're capturing exons or not, and capture coverage, how high the coverage is at those areas, which can increase our power to call things like SNPs. So first and foremost, how are we accessing this exon coverage and specificity? So we're going to take a global look at gene regions first, UTRs, exons, coding sequences within there, and then also some UTRs as well. So Tiny, this is a tiny depth profile for that small combination. On the x-axis, we have depth, and then the y-axis is the percent basis of depth. And each color of the line represents a different genic region. And we can see this really nice depth profile kind of decreasing as we get towards the higher end of things. And Tiny and Goldilocks really have similar depth profiles, even though they differ within their gDNA library size. So these are pretty comparable in terms of their specificity across genic regions. And also, Andre 300 has a similar depth profile across all of these genic regions as well. So it doesn't look like gDNA size is really having, a larger size is having an impact on the specificity. But when we have this mismatch in terms of larger probes and smaller gDNA with Biggie Smalls, it has a lower depth at many of these genic regions. And I can give you a little bit of an anchored reference point. And you can see that specifically the UTRs and genes are at a lower depth profile at that about 15 depth. So in terms of will longer probes capture divergent gDNA regions, our Biggie Smalls combination captured lower depth across those genic regions. So not a good option for moving forward. Next, we're going to be taking a look at assessing exon coverage and specificity only at exons, so discarding those other genic regions that we're looking at. And this is a little bit of a box plot for percent in exon capture. And we can see that our green box, which is Goldilocks, has a higher percentage for in exon capture. And that Andre 300, right, the 300-300 combo, has a little bit of a lower percent exon capture. And when we look at near exons, right, 150 base pair flanking regions from these exon regions, we can see that Goldilocks actually has a pretty good return, while Biggie Smalls is not doing well, and Andre 300 has a comparable percent near exon capture as Goldilocks. But really, it's this off-target region where we can see how this all comes into play, because our Goldilocks method is really reducing that off-target capture, and that's where we're seeing the gains in near and in exon percent capture as well. And so across these things, Goldilocks is really telling us, hey, I'm just right. Follow me. So kind of to sum this up, Andre 300 near and off-target capture is worse than these other 2 combinations that we're looking at. And so to lastly take a look at even a more zoomed in picture, we're going to be taking a look at HSP 70 and zooming right into that region. We have a kind of a piece of the oyster genome here on the bottom in a depth profile. Gene regions are in purple, and then the different exons are in different colors with coding regions in white. And we can see here that this is the distribution of coverage across this HSP for tiny. And when we overlay the depth coverage for Goldilocks, we actually see a more uniform and higher distribution of coverage at this HSP, giving us better power to answer questions about what's specifically going on with this protein and gene. So just to recap, Biggie Smalls is a big mismatch, big strikeout. Probes larger than gDNA size negatively impact capture specificity. Goldilocks is really best for in-exon capture and near-exon capture, and this is just a larger modal size invariance in our gDNA libraries, and that there's been really great gains in exon coverage across specific gene regions with this Goldilocks method with the smaller probes and larger gDNA. So really, EEC can benefit as a method from larger and more diverse gDNA libraries, so Goldilocks is the way forward. Well, thank you very, very much, folks. And I'm willing to take a couple quick questions. GitHub repos there for methods and any pipeline stuff you have questions about as well. Thank you.